I could start with myself. I'm an engineer. I have been working at Indiana University for 22 years. Our major research is in data visualization, and with HapMap, we are part of the Hive technology core, and I'm leading one of the mapping components. And Rahul Sahid, he's leading the other mapping component, and you just also saw him in the CCF session. So I think potentially we are looking at different people right now, but I think it's good to have us all in this together. And again, it's so unfortunate that we can't meet in person. It would have been so much nicer. So let's do a brief round of introductions. Bruce, Kirsten, if you want to go ahead.
Yeah, so I'm Bruce Herr. I work at Katy's lab, and so I'm from HapMap as well. So.
Hi, and I'm Kirsten Meyer. I work at the Sanger Institute, and I'm part of the CCF Lung Network, as well as Discover, which is a European lung consortium.
Nice to have you here, Kirsten.
Hi, I'm Kristen. I'm with NIH in Rockville, Maryland, and I work on the 3D models that are going into the user interfaces for Katy's group.
Hi, everybody. I'm Andreas. I am a Katy's PhD student. By the way, what you see behind me, this is a kidney model that Kristen actually developed. So,
Brian, Zorina Galabiana.
Sure. Hi, I'm Brian. I work at GE Research on the HubMap project there that's being led by Yosef El-Kafahi. I'm sorry, am I speaking over somebody? I hear other voices.
If you're not speaking, please mute yourself. Thank you.
Oh, okay. Thank you. I'll continue. My background is in computer engineering. I've been working at GE, working in healthcare and a variety of other things for over 30 years. I'm co-PI on an ITCR project having to do with software for tumor heterogeneity. I also worked with some other folks from GE that we released an open source software toolkit for visualizing images and running analytic pipelines on images. So, very interested in the topic of user interfaces, how images at different scales are displayed, how registered, and so forth, and different types of data visualization.
Thank you. Zorina Galabina.
Hi, can you hear me?
Yeah.
I'm Zorina Gallus, as you can see, HapMap. I guess my official title is coordinator. I have a background in physics, biophysics, medicine, pathology, bioengineering. I worked in academia, then in industry, and now I'm at the NIH. So I worked at the Heart, Lung, and Blood Institute. And I am one of the people who started HubMap.
Hi, my name is Galabina, and I'm a user experience researcher, and I work at the European Bioinformatics Institute, and we are part of the data coordination platform for the Human Cell Atlas. So I'm here to learn primarily. Thank you.
Thank you.
Hi.
Christine. Yeah.
Hi, I'm Kristen Ardley. I work at the Broad Institute, and I've been leading up the GTEx efforts.
Sarah and Amy.
Hi everyone, Sarah Teichmann, welcome Sanger Institute, co-chair of the Human Cell Atlas Organizing Committee, and really curious to learn more about thoughts on common coordinate frameworks.
Hi, I'm Amy Bernard at the Allen Institute in Seattle, Washington. I'm affiliated with Brain Initiative, with Kidney Precision Medicine Project, and with the SPARC project.
Thank you. Albert and then Galabina.
Hi, Albert Berger, head at University of Edinburgh, and I'm part of the Helmsley Trust Gut Cell Atlas Consortium.
And I think Galabina already introduced herself, but then that we have Chris Briggs, Elizabeth Record, Griffin, and Jennifer.
Hi, I'm Chris Briggs. I'm working with Niels Gellenborg's group at Harvard Medical School, and my role is Senior Data Curator for the HubMap.
Liesl, Griffin, Jennifer, do you want to briefly introduce yourself?
Sure. I'm Jenny, and I'm with Stanford. I'm coordinating the data for the seed networks along with CCI.
Thank you.
Hi, I'm Griffin Weber. I'm a medical informatics research at Harvard Medical School, and I'm working with Katy Borner on HubMap.
And we have Daniel Miller and Liesl Record.
Hi, I'm Dan Miller. I'm from the NIH. I'm part of the BICCN and the Brain Initiative.
Wonderful to have you here. Liesl, we couldn't hear you. So I will bring up the original slides again. Let me share my screen here. These are questions that actually were composed together. So when Jim and Peter first talked, we thought these anatomical structures and cell types tables, and I am happy to tell a little bit more about those, are very core to what we are trying to achieve in our respective consortia efforts. And while you are reading these topics, I also wanted to make sure that every one of you has a link to the Google Docs that keeps track of all the comments that are made. And I might even ask Liesl, if you are available to take meeting notes, that would be wonderful. I feel overwhelmed to lead the discussion and to also take meeting notes. So this would be wonderful. And I put in the chat log the link to the Google Docs that is supposed to capture our meeting notes. So before we can go to these sub questions, let me give you one more introduction, because there was a question of what are these tables like, and so I have another slide set here that I think will be helpful here. So as you saw in the 2 slides I presented, there is a deep need to take these anatomical structures, cell types, tables, and use them for ontology design, autonomy, which is used in our exploration user interface to just semantically filter out and zoom into certain subparts of the body, but also to then cross-link to existing ontologies, but then also needed for the 3D object design. And again, we have Kristen Brown here on the call, which actually was the expert who created these 3D objects that are now used in HubMap for not just registration of tissue cuboids, but also for the exploration of spatially and semantically annotated data. And the original table of different cell types and associated markers comes from a paper by Sanjay Jain out of KPMP. And some of you might have read this paper, and this is table 5 in this paper, which then for a while was just called table 5 within HubMap, but then ultimately got renamed into anatomical structures and cell types table. And here's an example for the kidney. So you get to see structures and substructures and then associated cell types. You also have subsets of marker genes in here, and you have comments as well. This seems to be a type of information that can be created for most of the organs that we are dealing with in HubMap. So when we asked our colleagues at the University of Florida to create such a similar table for the spleen, they were able to do this. And now other tissue mapping centers are creating other tables like this for other organs. Obviously, it's not just genetic markers, but it might also be lipids and proteomics and other ways that cell types can be identified. As you might know, inside of HubMap, we have 25 different assay types. So you can imagine 25 more columns here, which could potentially be used to agree on cell types. When we first discussed, so Jim and Peter and myself discussed trying to see if we could get consensus around these tables, Jim rightly pointed out that it would be a major undertaking to agree on these tables. Right now, I think I would be very happy if we would have major sub-regions, anatomical regions, and major cell types identified. It doesn't have to be complete, and it doesn't have to be perfect yet. But I think if we could see if other consortia have already created these kind of hierarchically nested structures, that would be very useful. So within HubMap, we are planning to have these tables for at least 5 organs over the next year, so by the end of this year. And again, this is very central for anything we do, because ultimately it is used in the exploration user interface. This is actually the table here, which you use to browse into anatomical parts. And then very soon.
Which 5 organs can you say?
Say it again. Oh yeah, so we have the table so far for the kidney and the spleen. And then University of Florida is also doing it for 2 more organs connected to the immune system. Then there will be the heart and the colon and the lung. So these are the organs which are next in line for HubMap. And on Kristen's side, and again, it's wonderful that you could join today, we have completed the kidney and the spleen, and we have that for male and female, and obviously there are 2 kidneys. So there are 6 total organs right now which have been modeled and are also annotated. And again, all of this is then used to support the registration of tissue cuboids, but then also given 3D object collision, we can have anatomical terms identified based on the collision of 2 objects, the tissue cuboid, which is a virtual object, and the 3D object. And Andreas Bückler, who is also here on the call, he has been implementing that interface. And in case you're interested to explore these interfaces, there are the links, and so you can go and play with these interfaces. So what I can do is to copy them over into the Google Docs so that you have an easier way to get to them. Ultimately, it's important also for you to understand that inside of HubMap, we do use a linked open data knowledge graph approach. And we have a way now devised, thanks to Bruce Hare, who is also on the call, to interlink spatial entities with spatial object references, and then place objects inside of objects inside of objects. So a cell inside of a FTU, functional tissue unit, the functional tissue unit inside of microanatomical structures, inside of larger anatomical structures, inside of the human body. So this ultimately gives us a registration that really goes from the cellular level all the way up to the body. But you can also spatially locate a single cell based on its placement in a cell segmentation map, based on the knowledge you have about the set stack of tissues, based on the registration of a tissue cuboid inside of 3D reference organ, such as the ones you see here on the right-hand side. So let me stop here and see if there are questions.
Yeah, so I have 2 questions. One was whether you could paste that bioarchive into the chat or into the Google Doc. But then the more profound kind of one is, how can we, in order like for that common coordinate framework to become kind of experimentally sort of relevant, you need to have a kind of reference set of experiments with some modality like spatial or single cell or something, where you know, like each, let's say you're talking about cuboids, you know where each cuboid is, and then you do the experiment for that cuboid, and then everything else can kind of be mapped back to that. And is that part of the exercise kind of that goes with the models? Because otherwise they kind of remain sort of theoretical models too.
Yeah, so inside of HubMap, So the way it works is there's a kidney which might be an organ transplant which didn't happen. This kidney is typically butterflied and a tissue cuboid is extracted from it, dissected. And then that tissue cuboid might be frozen and then sliced, and then different slices go to different types of assays, single cell assays. And then that data comes back. And so the tissue mapping centers have very much a process in place to keep track of all these different slices and what kind of assays were done to them. And they also have, in many cases, cell segmentation algorithms that they're applying to understand what cell is in what area of such particular tissue slice. But then given our registration into these reference organs, which you see here, so we currently have 25 tissue cuboids for kidney, and 24 tissue cuboid registrations for the spleen. And there are, for the spleen, there are extraction sites. So there are these bands, which you see here, CC1, 2, and 3. The same will happen also for colon. There will be 8 different extraction sites. For the heart, I think there are 9 different extraction sites. So sometimes they're predefined, sometimes they're not. And then based on the combination of the information from the tissue cuboid registration, the set stack, and also the information on where on that tissue slice certain types of assays were run and what the results are, you can reconstruct a 3D object, and oftentimes you have multiple types of assays applied to the same cell. Sarah, does this answer your question?
Kind of. I mean, in an ideal world, what you want is like a comprehensive gridding, so that if there's then a biopsy is taken from a place that's not as carefully registered, say from a living, you could then register it back. Do you see what I mean?
Yeah. So I think there's one is the CCF itself, right? So if there is a lung biopsy and the CT scan exists for the person from which that biopsy is taken, which happens in some cases, which is ideal, then of course you could go to the right place, and you could even sample it multiple times, and you could register it back to the lung, to the person from which that biopsy was taken. We don't have that luxury in HubMap. Currently, we are using the visible human from the National Library of Medicine as a reference organ donation source. So we have male and female there. And then we have kidneys from other human beings, which might be older, different body mass index, different ethnicity, different sex. So there is a non-perfect match between the tissue cuboid and the reference organ in terms of sex, ethnicity, age, gender. But over time, these reference organs and 3D libraries will be more resembling consensus organs, if you wish, just like what the Allen Institute did with the consensus organ for the human brain. And then the registration will be more effective and correct. Inside of HubMap, you might also have seen there's an effort to use the vasculature to align tissue across individuals, independent of their age, sex, and gender, and body mass index, etc. So this is another way of getting that more perfect. But right now, in the interest of not losing information on where the tissue cuboids came from, we have implemented that registration user interface so that anyone can basically learn that interface in 5 minutes and then register the tissue cuboids in a minute. And I think some have it down to 20 seconds per tissue cuboid now. We do not support biopsies right now because we don't have biopsies within HubMap, but we would be very interested to collaborate with teams that need that and have funding for it. Ultimately, it's programmer time.
That's great, thank you.
Katy. And I think Jim just joined us all, which is wonderful. Welcome, Jim.
Thank you.
Are there any other questions?
Just a question about tissue types. Are you looking to have eventually all of the HubMap tissue types supported? The GE team we're working on, skin, which is not one that you mentioned.
So this is actually a perfect lead over. So one big ambition I have, and just call me ambitious, but I will try this, is that we all could look together at this anatomical structures and cell types table. And this is a draft. So I don't think this will be completely complete. This is really Jim and Peter and myself and selected friends and colleagues helping us all understand what kind of organs different consortia care deeply about. I just pasted the tiny URL into the Zoom group chat, and if everything went well, you should be able to click on that link and see the table in front of you so that you have it in larger and you can also look at it later. I would love to understand if we missed major consortia. I would love to understand if there are other organs which some efforts care deeply about and that should be added. As you also see, we did some initial summing up of a number of consortia which care about certain organs. So that could be used potentially as a way to pick 10 organs which we might like to focus on first. But you also see that there are some consortia which are more cross organs than others. So there are some which focus squarely on one organ and do that very well and care deeply. But then there are also some efforts like HubMap, for instance, where currently we are looking at 11 organs and very soon there will be many more. So I think we have skin in here. And currently it would be H10 and TCGA. But if there are others which also deal with skin, so please annotate or add it to the Google form that is associated with this particular session.
Thanks.
That is probably as good a time as any for me to say hello and just check in, if that's okay, to make sure that everybody's happy and content. As Katy mentioned the Google form. Do please try and capture stuff as you go along. I see the scribes are furiously typing, so thank you very much indeed for that. The key here is to capture the most interesting questions. You know, how might we do this? How might we do these something else? That's really interesting, stimulated by your conversation. And then after you've taken a break, come back and start to see how you might respond to those. So do use the Google Doc if you're not already in there. Follow the link from KaiStore. The other thing I'm going to say is take a break at some point, perhaps in 10 to 15 minutes, mutually agree between yourselves, take a 10-minute break, have a breather, grab something to drink, because it's hard work being virtual. I'm going to pop over to the other group, but unless anybody's got any questions.
Thank you so much, Toby, for watching out.
Okay.
Katy, you know, I can't modify the Google Doc, I can only comment, but I know of a lot of different consortia, additional consortia, like for instance, MRC, EU, and so on that I don't see on your table, and then also different tissues that I can fill in, but I can also do that offline with you.
I would love that, Sarah, and it's such a pleasure to meet finally. I have been following your work quite extensively.
Sarah.
And I really enjoyed the morning session also on the COVID work you and others have been doing together. Very impressive.
Sarah, forgive me. I just want to make sure that you can edit it. I'll see what I can do. I'm surprised you can't. It should have total. You should have total edit access.
I think it's my fault probably. So let me go there myself and change the permissions. Sorry for that.
Sarah does not refer to the Google document where we collect notes. She's referring to the table.
No, I'm referring to the one that was pasted into the chat.
Yeah, the table.
And I believe I have master. I have total. Yeah, this is me, and I think I just fixed it. Could you try again, please?
Okay.
So just so I'm absolutely clear, Katy, are you using a separate Google document then to capture the?
It is a spreadsheet, yes, and I think the link to the spreadsheet is in the chat, and it hopefully is also in the meeting notes so that other people can get there.
It looks like I can edit, so I think everybody else should be able to edit it as well.
Wonderful, thank you.
Yeah.
Yeah, please add.
I mean, I don't want to mess up your, I mean, do you want us to just add?
Yeah, please. I can
okay.
always compare it with the original.
Okay, fine.
Jim, do you want to add to this discussion?
I don't have any specific things to add. I mean, you provided an excellent example of one registration user interface. At our subtopic, we were going to talk about, the original idea was to talk about additional workflows for different types of data. You mentioned, for example, you're currently not considering biopsies. I think Peter is talking about a situation where they do have image-based data, so sort of image-centric sorts of localizations, and so there are general questions in terms of challenges in terms of quality assurance and quality control, in terms of being able to, at a minimum, be able to describe sort of the precision of the localization information that you're attaching to your samples. So in the case of what you showed, it seemed like there was perhaps slightly less of a challenge than those who are doing extremely high resolution imagery, where they're literally registering to sort of like voxel-wise degrees of precision, to situations of biopsies where it's really quite rough in terms of where one is localizing the sample. So one question is just sort of like in all these sorts of situations, you know, what is adequate in terms of being able to describe localization information?
And in our world, we distinguish the rough anatomical registration, which the registration user interface supports currently. And then there is a stage 2 registration, which is the biomolecular registration after the assays have been run and the data comes back. In our world, this is Globus and the Hive infrastructures at University of Pittsburgh and CMU together implement. In that moment, you then have the single cell information, in many cases spatially explicit single cell information. And if you would have a reference system where you know the composition and the distribution of different cell types for the different smaller and larger anatomical structures, you can then match your dot pattern from the tissue cuboid to the respective dot pattern in the reference organ. And for that match, there exist actually quite efficient machine learning algorithms, which can then in that moment morph and warp the tissue as needed to actually fit that reference organ. We are not there yet because we don't have that data right now, and we definitely don't have a reference organ that has that kind of level of precision. But we are very glad that we recently got KPMP funding to actually work with Seth Winfrey and Sanjay Jain's team on showing how that might work.
Ati, I have a question. As part of the table, do you have any information if any of those consortia or tissues are using specific ways to localize cells or CCFs or things like that? I don't think I see it.
I think it's very different from organ to organ. So for the
but the question, I'm not asking you to answer the question. I'm asking you if the table contains that information for various tissues or consortia.
No, I think it's different per organ and it's different per consortium.
So I think it would be nice to know.
For brain, we're doing it, but brain isn't currently on the HubMap list. But for brain, it is actively being done, yes.
So I think it would be good to capture that also in your table.
I like that, yeah. So I will try to add and maybe comments.
And so that in fact does present one of the challenges, because one of the big challenges, because even within a single consortia, there are diversity of methods used to map the same type of data.
Correct. So that would be nice to know.
Yeah, and indeed how to reconcile sort of the differences. Katie, you mentioned, for example, using machine learning to do mapping in your particular application. I can imagine a different team potentially using a completely different technique to map the same kind of data. And then the question is sort of like how to reconcile the localizations, the two different localizations that you get for the exact same piece of exact same sample, just simply because the two teams have used a different mapping technique.
Can I ask you a question going off of what Jim said? I'm sorry I came late. So I'm a cardiovascular pathologist. I've seen hundreds of autopsies, thousands of tissues, and I know at a certain level, at the functional unit, at a glomerulus, at a crypt of a colon, it all looks the same, and at a large gross structure, it all somewhat looks the same. There's some variability, but in between, it's completely random where a blood vessel is, where a nerve is between two people. You cannot predict based on one glomerulus where the next one will be next to it. So in that intermediate space, how are you making that a common coordinate framework?
Great question, and I would love to put Griffin Weber on the spot if I may. I don't know.
Yes, I'm here. That's a tough one. So we've been looking at something called a vascular coordinate framework, where we have a coordinate system where one of the axes is literally the pathways of the blood vasculature. So instead of looking at X, Y, Z coordinates from some reference point, you're describing a blood vessel and the position of that vessel, the distance away perpendicular from that vessel, and like a rotational angle, so that you don't have to specifically say this is the glomerulus at 10 centimeters over and this amount high and over. You can say it's just this is a cell that's 50 microns away from a glomerular capillary, and you don't really care where that capillary is across different people. You're aligning all those capillaries together, and you can describe positions relative to that. So that's sort of a simplification. Rather than an individual kidney, instead of thinking about it as 50,000 separate glomeruli, you're thinking of a kidney as represented by one glomerular capillary and the types of cells that are surrounding it. This is still kind of early stage. We haven't proven yet that this would work, but it seems like a way of taking advantage of the fact that most functional units are about the same, and the real difference between individuals is more the number of these functional units that you have and the 3D position of them. And by abstracting that away and looking at aligning things at individual blood vessels, it might help minimize that amount of variation between people.
So to follow up, just so I clarify that point, the vasculature at that level is quite variable as well, but you're saying simply the distance from the vasculature is what you're focused on, not the location of the vasculature. Am I getting that?
Yeah, we're saying that rather than specifying like the true X, Y, Z position in 3D space, you're specifying a blood vessel and distance relative to that vessel. So it would be 50 microns or 3 cells away from the basement membrane of the halfway at the midpoint of the capillary between the where it enters and exits the glomerulus.
So it sounds like that's fixing it to a taxonomic definition where your vasculature is a reference framework. Is that correct? Because you can have a spatial fixation or a taxonomic fixation point, and you're anchoring on your vasculature for the heart, because that doesn't work necessarily for things for which there's variance that requires a spatial fixation point.
That's correct. When in what method I'm describing, you're losing. If the kidney is reduced down to a single glomerular capillary, you're losing the differences between the top of the kidney and the bottom of the kidney, in a sense. The idea then you can split these things up eventually. So you can say capillaries from the one pole of the kidney versus from the other pole of the kidney. So you can divide these up as granular as you need to. But ultimately, you're going to be merging a bunch of vessels into one thing and losing some amount of 3D position.
But even if you
Yeah.
actually map it this way, you know, ultimately, Amy, you have to put it back into the body, right? Even if your map looks as crazy as this one figure from the
this is sort of the exploded map where you're imagining the root of the aorta is the center point, and then each vessel that goes out to individual organs loops around the functional unit, heads back as one of these different radial branches heading out, and you're putting all the functional units and capillaries around that outside ring, arterioles in the middle, and arteries and veins are near the center of this picture. So this whole thing, you can imagine rolling it up into a 3D structure, the same way that the human genome, within the nucleus, the DNA is all wrapped around in a 3D shape, and you unwind it into a linear sequence of DNA bases. But for analysis, if we had to always think of the human genome in this 3D structure, it'd be complex. By flattening out, you're eliminating the 3D position of it, but you're making it easier to describe the position in that simplified framework. So here it's the same kind of thing. It's exploding out the blood vessels into this radial pathway, but in that, how do you want to flip to the next slide? The next other figure. Using additional information like micro CTs or other things, we could ultimately take that linear vascular CCF and roll it back up into its 3D position within a kidney. So it's actually going the opposite way. It's starting with a kidney and they're tracing the 3D pathway of a blood vessel, of like a red blood cell as it goes around there, but straightening out and aligning it all the way on the right-hand side, which is a single line as you're progressing through the vasculature.
Yeah, that makes a lot of sense. I just wanted to specify that it wasn't, at first I thought you were talking about a heart-specific approach as opposed to framing it as a universal frame for how everything can be mapped back together.
Right, this is, we're talking about a universal method of describing position across any organ. You're actually, this is not like, you know, the heart here is actually the muscle of the heart, the cardiac artery, the arteries kind of going around. It's not the internal of the heart, which is just a bunch of blood. So you know, this is not modeling the heart. It's modeling the tissue surrounding the vasculature in the body. And you may not be, you know, when you're thinking of lung, you might think of the airways. When you're thinking of the kidney, you might be thinking of different structures in there. But what the vasculature does is it gives you at least a common way of describing location in that semantic way that you mentioned across any organ of the body. And that might give us a way of using kind of a common language across organs, as well as making it a little bit easier to align structures across different individuals. Since at that like functional unit level, people may be more similar than at the whole body level.
This is Zarina. I wanted to just give a much simpler, maybe, answer to Mark. And I think the idea of using the vasculature is the fact that each tissue has optimized physiologically the amount of oxygen that's needed by every cell and cell type in that tissue. And that distance from a capillary is never over 100 micrometers. And I think we are getting now some experimental evidence for that thing that's kind of known. So this is true for any tissue, but the actual distance depends on tissue and cell type and their metabolic needs. But it's safe to assume that you will never go, you'll have to go more than 100 microns around blood vessels to be able to define most of the cells in the tissue. And so the idea is that you can try to focus on differences, or you can try to focus on similarities. And I think it's easier to start with what we think is similar. And you as a pathologist, I'm pretty sure you can tell if it's a kidney or liver or a spleen based on patterns. You can tell if it's normal based on what you see and what's different than the regular pattern. And the pattern is very specific to tissue, and the vascular pattern is very specific to tissue. While it exists everywhere, it's a way of telling where you are without even knowing where you took the tissue sample from. I don't know if that is another answer.
No, that's fine. It's an interesting approach, and I'd seen a presentation on some of that vasculature before. I think I'm getting my head around that it's just a distance from a certain size vessel within an organ, and that's what you're calling the framework, which is interesting. I don't know how one zooms in and zooms out. If you envision starting big and then getting down into the nephron.
But the vascular itself zooms from the whole body level to the single cell level, because it goes from the largest, longest probably organ, the aorta, and then it goes to single cell level capillaries.
I agree. It's just so variable where you will see those vessels within a tissue. I agree with you. You will see the same cells around certain size vessels, but where you will see those vessels in a tissue, you cannot predict that. That's, I guess, my point.
I would challenge you on that, but we can take it on the side. You're close by.
All right, while Zarina and Mark are battling, we should all have a break. Maybe we can watch the two of them, but come back in 10 minutes.
The battle of pathologists.
So I will stay on. I'm happy to chit chat with anyone. I have my carrot juice right here. So please do bio breaks, do everything, but come back or check out what Peter's doing in his session. Feel free. But I'm looking forward to discuss more in 10 minutes. So it's 2:50 now. Let's be back at 3 p. Eastern Standard Time, summertime. Please come back.
I'm happy to stay on and chat, Katy, and also Kirsten, if you want to talk. I think Kirsten is also there and has a question.
Please,
if you want to stay on quickly. Is she there or no? Maybe not.
I was just wondering.
No, you are. Yeah.
That, I mean, mapping to the vasculature and is clearly very important, but I wonder whether in the short term there might be more insight from actually mapping exact neighboring cells in terms of cellular function. But you know, that's just a gut feeling, and I just wondered what other people thought about it.
I would suggest that we have, if we're going to talk more about that topic, would it be polite to wait till more people are back? Just because I feel
no,
like
I was actually going to discuss something completely different.
Yeah, I know. I was just thinking like if there's a different topic that's not right on this one, because it's such a good topic, Kirsten, that it's
a great topic.
No, because Kirsten basically sent me a question about her chairing her session tomorrow. So I think you've actually done a great job in how you've organized this one. And it's actually given me a lot of ideas for the session that I'm doing tomorrow, because the previous session that I was in, like, it didn't have any of the presentations or the Google Docs or anything like that. But Kirsten, you were asking basically about whether someone else could co-chair with you, I think, tomorrow.
If you had.
Is that right?
Yep.
But basically the way I interpret that topic is not that it's computational methods. So Kirsten's is on integration of cell annotation across modalities. Is it?
Yeah.
But does it refer to the computational methods, which is like more the nitty-gritty type of stuff that like Vitali and Mika and so on are doing? Or is it more about the annotation of the cells, which is more like cell ontology? So
I guess it is more about the annotation, but the point in a way is the annotation across modalities will rely on how do you integrate the data across the two, right? Because I suppose it also boils back to what do you do when you have a cell type that to all intents and purposes looks very similar functionally, but you get competing data. But I'm happy when I'll share the session to invite people's ideas and, you know, just take it from.
Is that one subtopic within a larger topic?
Yes.
And what is the larger topic?
Just cell annotation.
Ah, okay, now I understand. Yeah, no, because John wasn't registered, so I'm just trying to think. Rahul could help. Is he there?
He was here for a while. I don't know.
Rahul Satisha, Kirsten.
Okay,
he's registered and he's the developer of the Surat CCA for mapping across modalities. So he could help you.
Yeah.
If he's not busy with another one. Christine can send you the list of all the chairs. Christine Schwenk or Tracy Andrew.
Yeah, sure.
Yep. And then you'll see who's free in your slot and who might be able to help you.
Kristen just joined. And she's asking, what is the question?
Oh, Kristen, hi, Kristen. So Tracy's, Kirsten's question is whether Rahul Satisha would be free to help her co-chair her session in the slot on cell annotation tomorrow.
It's Kirsten Meyer or?
Kirsten Meyer, yeah.
Yeah, let me connect to Kirsten with Raul. Sound good?
Yeah, because he was in this session and then he flicked out.
Ah, okay, let's track him down. I'll find him.
Very good. And there was a question of if I could share the slides I presented. I'm happy to do this. I have to find out how to do this officially. If you could add your names into our official Google Doc, that would be helpful because then I can make sure you get an email with a link. So we have to find our Google Doc again. And Liesl, thanks so very much for scribing down while we were discussing. It's actually a really good idea to have somebody helping with this. I didn't realize that I can't do this alone. And then Sarah, in your world, have you seen these table 5s or these anatomical structures, cell types tables? Because again, they could have many columns, right? There could be a column for genetic expressions of different cell types. There could be a column for different stains that are used to identify different cell types. There could be proteomics and other assay types that could be used to identify these cell types. And so I think this must be something that almost every consortium needs to do.
Yeah, so I wanted to actually catch up with you to chat. I mean, now we have 5 minutes, but maybe another time also offline. Hello? Yeah.
Yeah, I hear you.
So to just catch up in more detail, because I have to say I didn't follow this table. Yeah, so exactly. No, so this is exactly the kind of thing that we're doing with cell ontology.
Okay.
Kirsten, this is kind of what I meant by pathology and cell ontology.
Yeah.
Because basically in the NHS, so the way we were approaching it was that, you know, cells, and this must be, I don't know, probably similar in the US, is that there are certain antibodies that are used as, conventionally used as markers for clinical studies, you know, for clinical pathology and histology by in the UK, you know, kind of standards by the Royal College of Pathologists. And what I want to do was do this, exactly this kind of thing, coming from both the single cell genomics perspective and coming from the medical kind of antibody conventional immunohistochemistry perspective. And then try to link essentially the basic science with the clinical world of pathology. So we have our kind of cells, and we're naming them new things, new cell states, and we have new marker genes and so on. And but then there's also the clinical world that has their kind of conventional, you know, immunohistochemistry sort of antibody markers that they use for specific, you know, cell types that they've defined for decades and so on. And I wanted to kind of, and so we're working to sort of bring those together And I was wondering, you know, whether it makes sense to link on this and also more on the coordinate thing. I haven't filled out the whole Google Doc, I should say. But you know, the MRC and EU projects are publicly available. I'll send you the and Tracy, I think she's not online here, but she has an overview of all the tissues and all the awards as well, Katy, for.
This would be very helpful. Yeah, so I'm making notes here.
Yeah. So if you go to.
Or you could put it in the chat link.
So Tracy Andrew, yeah. So you can, I mean, these are all available online, like publicly available, but Tracy Andrew has she's the HCA executive office. Oh, sorry, no, I mean, sorry. I sent this to you, Kirsten, by accident. I meant to send it to everyone. Oh, sorry. Getting close
Sarah
to
and everybody, I think it's not a good idea to start with all these organs. I think right now we have 38 minus 3, 35 different organs. I think that's too many, but we could potentially start with 5 to 10 and just try to get these anatomical structures, cell types, tables for those. And again, these tables don't need to be complete. They can be partial, but we would then be able to agree on ways to share this, on ways to officially approve them by anatomists and pathologists and others that are subject matter domain experts on those organs, which I'm not. And then also try to share back that information with some of the other efforts which are caring deeply about these organs and see if they would like to use that, or if not, why not, etc. I think that would be
I think it makes a lot of sense, and that would be fantastic. And I've put in the chat Tracy's details, Katy.
I got it. Mm-hmm. Totally got it. And it's 3:01, and I'm German, so I'm going to restart this all again. And so let's see whom we have here. Let me find, stop sharing, and welcome back, everybody. Good to see you all back. And we will start with Kirsten's absolute phenomenal question. And Kirsten, do you want to restate it? Because I think we have some new people here which might not have been here when you first asked that question.
Great. Okay. Well, welcome back, everyone. So I was just wondering, I think it's fascinating the way the efforts that have been made to put organs into that larger context of, you know, the vasculature and all that. But in terms of actually gaining biological insight, I was wondering whether in the first instance, really looking at a smaller scale and just trying to understand who are the neighboring cells for any given cell, might that be in a way more an easier way of really beginning to understand the biology of individual cells? And again, I have no predefined view and was just wondering what other people thought.
And I can do a quick answer, but I think there are other experts on the call. So one of my PhD students, he is working on not just cell segmentation, but functional tissue unit segmentation. And so we are working on data actually from the Human Protein Atlas on glomeruli. And then you can identify what kind of cells are close to what other cells and how far are cells away from the blood vessel system inside of just a functional tissue unit. And actually starting with functional tissue units, which are doing the heavy lifting of what needs to get done by the different organs, seems to be a good idea. So I would be very interested to hear from others what they think about the idea of actually using functional tissue units initially to test out that approach that Griffin presented. And by the way, if you use the vasculature as a reference system, there are some cells in our body which are touched also by the lymph system and or the nerve system. So you have two other systems to cross-validate, and as an engineer, I deeply appreciate this. So if there are two cells which are served by the same blood vessel system, they should have an address that's close by. Again, if they're also served by same lymph or same nerve cell system, they should also be close together. If that isn't true, then something went wrong. And so I really like that we have this triangle cross-check, not for all cells, but for quite a number of them. So I will leave it here.
Yeah, just a question or a comment on that. It's very interesting with the three different systems. Would there be value in considering the skeleton as a fourth system? Yeah, sort of like where in the body is it?
I see it more as the stem, you know, where you hang it all back on together like a Christmas tree, which you're trying to document. Because basically what you saw in the conceptual drawings that Griffin Weber explained, this is a map which nobody has seen like this before. Actually, people started to do it by hand like 60 years ago. Griffin found some fascinating early work on this by another gentleman named very similar to him. But it's now that we can do it in real with the single cell omics. And so ultimately it will look very different. It will look very alien, very artificial. And at some point you will want to bring it all back into the human body so that it looks again like a human body. And I think that in that moment, the skeleton can be very, very important. But for creating that reference system, I would say no, but I'm just one out of many experts here.
Mm-hmm.
Might this be a good time to break in? It's Toby again, just making sure everybody took a break, stroked the dog, got a cup of tea, whatever it was that you chose to do. Point out that it's probably 60 minutes until we break formally at the end of the day. We'll break at about 4:05 Eastern Standard Time, or Eastern Time now, so it's summertime, isn't it? And please don't forget, if you are so inclined to continue filling in the Google Doc that you access through KaiStorm, that's where you're capturing in the blue section some of the key thoughts you have about any of the collaborations that might work across consortia. So that's the place to capture that. It would be lovely. It would be lovely if we were able to generate some really new and interesting collaborations between consortia as a result of this fabulous conversation led by Peter, Jim, and Katy. So forgive me
Toby,
interrupting.
can you remind us what happens after this? Do we go back to plenary, and what happens in that plenary?
Yes, and we don't want to bore you in any way. This is the most important and powerful part of it, is the conversations between really, really bright colleagues. The intention is that we come back very briefly just to say, what are we going to do tomorrow, but critically to share the data that has been created so far. Now, as we said right at the beginning, it's just impossible for every single person to feed back. So what we're inviting you to do, and this is why it's important to fill in that Google Doc, is the blue section. That's where there is a description of the collaboration, the potential collaboration between consortia. If you are able to fill that in, if there's a project that you think, yeah, that might be useful to take forward, we'll capture all of those and put them into a gallery so that everybody can have a look at them, comment on them as you see fit. So that's all we'll do at the end. Quick wrap up, show everybody the gallery, and remind you when we're starting tomorrow.
Brilliant, fantastic. Thank you very much, Toby.
And Toby, Toby, I really enjoy also your accent, and I almost expect the 3 o'clock tea, you know, the high tea. It's just wonderful to have you. Thank you.
I haven't. Why didn't I bring my tea with me? Tomorrow, Katy, I promise you, we will have tea together.
Thank you so much for reminding me of good old Europe.
Very good. We'll see you later.
Yeah.
Don't work too hard. Take a break. Fill in the document. See you in a bit.
Okay. So given that Toby just gave us marching orders, if you could please go to the Google Doc and add your names to the list of attendees at the very, very end of the document. Let's do this all for a minute. So there's a list of attendees at the very, very end of the document. Liesl Record kindly posted the link to the Zoom group chat. So Sarah, can I ask you, how do you agree in the UK on the pathology and cell ontology tables? And are there papers that might describe some of those tables for organs, including maybe also the kidney and spleen, so that we could compare, but maybe also for other organs?
So basically, we've been discussing this with the cell ontology people at EBI. And so we have, so there are a couple of things. So on the pathology side, from the clinical side, we've got tables for various organs. Now in general, that we just we've just been collecting internally. I don't know if the Royal College of Pathologists has those like published somewhere, but it's basically what the pathologists just give us for each organ. Often they're more coarse grained than what the single cell genomics and spatial approaches discover in terms of cell states, and the markers may be different and so on. And so the question is, and then cell ontology is different again in the sense that it's very fine grained. I don't know if you're familiar with it. It's very fine grained for some like parts of the cell space, I'll say, but then less detailed in other parts, just because it's been, I think it's just historical accident, the way it's been populated over time has been patchy, and just wherever funding is available or wherever there are projects that are going on, that's been done in detail. And so what I want to do, and actually I just sent an email earlier today, is to link up with them to have some way of recording within the cell ontology database these different layers basically, and harmonize it within their ontology. But I don't know. Yeah, I mean that's my kind of plan.
And if you have papers that might publish partial tables, for instance, maybe just for subparts of an organ, or that do it for the gross anatomy, but not linked to cell types, I would still be very interested in seeing this, especially if we can expand it to capture from the gross anatomy to the microanatomy, functional tissue units, to these cells. And then again, there are 25 different assays in HubMap which try to get to different cell types and states. And there's much discussion what that even means and how to call it all. But again, if we could agree on some major types of cells and then the subtypes, and then bring this all together in these tables, which are easy to read for the domain experts and can be analyzed and approved by pathologists and anatomists, I think that would be a major step forward for us all here.
Yeah.
Can I ask who is coordinating those maps for the lung within HubMap?
So Rahul has been working with Gloria on the lung, and please ask him. I have not touched the lung because that was what Rahul was focusing on.
Okay, thanks.
But I think with COVID, there's a much need to have a better understanding of how the lung works. So I could also imagine that there will be much, maybe not biopsy, but autopsy data becoming available, maybe not of healthy, but of diseased lungs. And who knows, maybe there's also more will to open consent to having biopsies done, because everybody on this planet will understand how important that research is.
You mean biopsy from the post-mortem, warm autopsy, or?
So I could imagine that there are family members that because they have lost loved ones, that they would be more open to be part of studies that aim to make sure that nobody ever loses a loved one.
But you could, I mean, that's clear. But I'm just trying to understand how you distinguish biopsy from, let's say, taking the entire lung out and then dissecting it. So I'm just trying to understand how your definitions of
So
the biopsy versus the.
Can I jump in real quick? So I've been involved in a little bit of this. I presume you're talking about COVID related autopsies. So up to date, most of the autopsies have been with biopsy. So rather than doing a complete autopsy, there have been small pieces of tissue collected. That's going on in China. Certain countries of the world won't allow autopsies on these cases. I've been involved in multiple discussions over the last few days. In the United States, OSHA put out a statement saying they didn't want people to do it. Then they kind of backtracked. And the CDC is now actively seeking out people to provide tissues for them to do analyses. So this is ongoing at multiple institutions, including.
You might have lost
actively seeking members to who also do autopsies to participate. So if there is interest from HCA or some other group that can do something sophisticated, feel free to reach out to me. I can make some connections potentially with people who may be doing autopsies in the future.
Also, I wanted to say that I think kidneys are also a key target organ in COVID.
Yes, kidneys, obviously lungs, but kidneys and hearts are also interesting, as you saw earlier about ACE2 expression and renal failure and heart attacks, or at least increased troponin levels in hearts.
I mean, and GI is kind of like very obvious, isn't it?
Yeah.
Large and small intestine, because the enterocytes are very high in ACE2 and 30% of the
But
patients.
I think it's thought that the virus that's in GI is not necessarily live or infective. I thought.
But there is an enterocyte population that has very high ACE2 and TMPRSS, so it is.
Yeah, but if the virus is not functional, it may not matter. I don't know. We are all sort of making more sort of assumptions than knowledge.
Yeah, I mean, for here, the reason I was asking is just that we have, so we in here in Cambridge and kind of the Wellcome Consortium has a tissue source that's the Cambridge or Collaborative Biorepository for Translational Medicine, which is the transplant surgeon team.
So for instance, I saw there was a difference in children. They think that actually that's why small children are not affected, is because they are more likely to excrete, to pass the virus. Also, it can be detected in stool, but I think from what I've seen, very few papers, it's not clear that that's actually a live form of the virus.
Mm. Yeah, there's obviously a difference between just viremia detected and infection. But anyway, can you hear me? What I was saying was that a lot of our tissue source is through biopsies up to 5 centimeters from that are accessible to the transplant incision, which goes from the neck to the waist. And those are the fresh tissues. And now we're also applying for rapid ethics for warm autopsies, which are then, obviously a completely different procedure, which is after, it's not transplant donors, but diseased patients. And my impression is that basically it's just done with full PPE, the autopsy, and that it's not a major. It doesn't, that there's not a huge hurdle to that. So I'm surprised to hear that it's such a big issue in the US, actually.
Yeah, so in the United States, one of the concerns is using PPE, which is somewhat limited at many institutions, to quote unquote waste it on a deceased person versus having enough PPE for nurses and doctors.
I see.
But you're right. In England, it's classified as a level 2 autopsy, which is, there's 4 levels, so that's not a very high risk autopsy. It should be done. It's going to be done at lots of institutions, but some will say no for PPE purposes.
I see.
I think this discussion is very important because most likely also for HCA, there might be implications of COVID on the amount of tissue that can be collected over the next year. But I think some tissue we might actually have a lot of, and others we might not have much because many of the labs are closed.
Absolutely. Absolutely.
All right, so we could go back to our original questions. And if you have all the Google Doc in front of you, you would actually see in the very beginning the original questions. So we have scribe captures notes, how might be. And again, thanks so very much for everybody who added to this part. If you would like to add more to this how might we question set, please do. If you have links to relevant papers that we all should read, please add them here. If you have links to other efforts that might like to get a copy of the slides that I presented initially, or that might like to get pointed to that big table which we are compiling together, please add them here as well. And then the original first set of questions was related to major anatomical terms and 3D structures in human. I would like to know from the people in the room we have here, if there are other efforts to create 3D object libraries, like what Kristen Brown is working on. Are there other efforts to have basically 3D models of reference organs, which are anatomically annotated, ideally with crosswalks over to ontologies, just like what we are aiming to do? Are there any efforts on that?
Hi, this is Albert Berger. So we're early days yet, but for the gut, which is the gut cell atlas, yeah, in Edinburgh we're planning to have 3D images of gut and trying to connect that to what at the moment is a linear model of the gut, together with some anatomy terms. So there is, and there's a third part which TBI people do, that's to do, which is so-called anatomograms, which are depictions. They're actually 2D, but you can go into more detail, and probably Sarah can better talk about those. But yes, so part of our program in that context is to try and reuse some of the work we've done previously in some 3D mouse atlas to have a 3D gut representation and map that into the sort of coordinate framework for gut. But it's early days.
And Albert, if you look for your name, you could also add links to papers or websites or whatever it might exist. I would love to check out the 2D anatomographs. I love the name already.
So I asked somebody else to do that because I'm not involved in creating that. So these are colleagues at the EBI, and they have done it for other organs, but they're now in the process of developing them for the gut. If nobody comes forward and puts them in, I'll do it, but it's not my work. We just want to use the anatomograms to support the representation and the localization of the cells in the gut.
Do you happen to have a slide with an anatomograph so that we get to see it?
Anatomograms. I can look it up. So if you want to carry on with the discussion, I'll look it up meanwhile. Maybe I'll find it.
Sorry, Albert, can I just check? When you say, so when you say anatomogram, because there's the 2D kind of representations, but that's not what you're talking about, right? You're talking about the full 3D reconstruction.
Well, let me find a slide, because we recently got a presentation from colleagues at the EBI about the work they do. Is nobody else there from the EBI at the moment? Because I saw.
I didn't see anyone in this, to be honest.
Hi, I'm Gabby. I work on the DCP, but I'm definitely, it's not part of the DCP team, so I'm not sure actually what you're referring to. It might be part of the Expression Atlas.
I think he means the single cell atlas, like
maybe it's the Expression Atlas.
Yeah.
Yes, they use it. So this is Irene who is our partner.
Yeah.
I'll dig up a slide and I'll show it, but in the meanwhile, maybe you want to.
Okay, no problem.
Thank you so much. And then the other next session was all on calling cards, as I call them. So these are cell type calling cards. And again, these are the other columns in that big table we have been looking at. And they could be based on antibodies. They could be based on stains. They could be based on other assays that can be done at the single cell level. And I wonder who else is working on calling cards. I know Peter Kotchenko is leading an effort, and we have been in recent discussion, but I wonder if there are other efforts that are trying to get us so-called calling cards for different cell types. There must be efforts out there because it's core and central to understanding what cell types exist where in our human bodies.
So we're working on basically logistic regression models of cell types, but I think that's slightly different. So you mean like with respect to the coordinates of where they sit within a coordinate framework?
So ultimately, I would love to have these cell calling cards as additional columns in that big table. So right now we have gross anatomical structures, microanatomical structures, functional tissue units, and then we have major cell types and then single cell types. I mean, cell types at the most specific level. But now the next part is to understand how to actually identify these cell types. And so that's really expanding the table outwards to the right. And again, some cells might be easiest identified using antibodies. Others might be perfectly fine identified just using stains. Others might have other ways of being identified as that particular type. So I'm looking for a lookup
like
table.
a multimodal. Like
to just, basically trust you guys that you got me the right calling cards, but then I can work with these calling cards and I can develop interfaces that really identify the right cell calling or the right cell types. And they also give you a reason of why I did this correctly, because I point to that paper or I point to that effort, etc.
I mean, so the perspective that we are coming from, like in the first instance, is single cell RNA sequencing data for the kind of broader sort of reference. But then you're completely right. I mean, I can see what you're saying, that you want to make the description deep with morphology, sort of H&E morphology, or cytospin, or you know, electron microscopy data if you have it for that cell type, or protein is obvious. Yeah, I can see what you're saying. I mean, I think that will take time to get that complete.
And right now, I don't think we need it complete, but we need to get people to look out for these kind of cell calling cards, because again, whenever you have a new one, you just plug it into the framework we are now using within HubMap. And then if that particular result comes up in all of the many assay types, then we can say, oh, we found this pattern. This must be a cell of that particular type in this particular universe, because there are different views of the world, obviously, and they might name these cells slightly differently, or might think that is a state instead of a type.
So Amy has just said Brain Initiative is working towards this goal. Maybe you want to elaborate on that, Amy, because I know that Brain Initiative in a way is, or especially Allen Institute and so on, is really far advanced on this for the brain. Is that okay, Katy? I know you're the chair. She's just put it into the chat.
Oh, I see. I didn't see that part.
So I also put in a screenshot from one of the anatomogram slides.
And I have to download it. Let me do this. Do you want to screen share and show it to all?
Yeah, exactly. So this is what the illustrator in my group is doing, Albert. This is what Jana is doing. Yeah. I mean, this is, yeah, I mean, this is basically sort of our common sense kind of breaking down to the level of resolution where we can essentially sort of place single cell RNA sequencing data based on sort of landmark gene expression, bits of spatial, and so on. And we're doing this for a lot of organs. And then essentially sort of placing.
It's another way of describing location, and
yeah,
I think it's sort of something complementary to all the other ways of capturing that.
Yep. Yep. No, thank you very much, Albert. That's brilliant. Thanks for pulling that out.
So, but this is 3D?
Well, kind of.
Okay.
The representation is, I mean, the meaning is in 3D, but the representation is in 2D.
Okay.
That
Yeah,
makes sense.
because I think that's what Katy was asking, right? 3D?
Yeah, ideally in 3D so that we can do annotations, the semantic annotation, and also the registration based on collision. So just like in video games, you can actually realize that you're running into a wall or you're hitting a button, etc. You can actually use the same collision detection when you register a tissue piece into a reference organ. You would then automatically realize, okay, that tissue piece now overlays or intersects with these 3D objects. And therefore you can semi-automatically annotate your tissue pieces when you register them. And so we are very interested in 3D reference organs, and that's why Kristen Brown here on our team has
Sorry,
been spending.
I've seen some of the 3D organs. It's mostly related to heart and lungs and other things that are now being produced by various pharmaceutical companies, even to have sort of patients fly through the organ and look at things. So I don't know if that qualifies for what you're asking.
Yeah, so there's, for instance, Atlas 2020, which you can download on your phone, and it's amazing. I mean, they did a really fabulous job in modeling the human body in 3D, maybe not perfectly anatomically correct, but just beautiful for understanding how nature parts work together. And they even have some animations of muscle groups, etc. So I think there is a lot of 4P material, but I think NIH would like to create a free resource which all of us can use. And so I'm trying to understand who else is working on free models, because it's very time consuming and it takes a lot of expertise to create just one organ model. And now we want it for 10 plus organs. So combining sources and resources there would also be very valuable on the HubMap end.
And these kind of anatomograms that Albert showed that we're using for database, for the single cell expression atlas visualization is more, it's not registering a tissue chunk or a biopsy or whatever in 3 dimensions. It's more placing the cells down to what you call functional tissue units, I think.
Yeah.
So, let's say it's a loop of Henle, but it could be anywhere in the kidney. But it's an epithelial cell that sits somewhere in the, somewhere in the loop of Henle, let's say, but that we can say that confidently, but it could be anywhere in the kidney. And what I would be curious about is whether you can actually tell a difference between a cell that's in one particular glomerulus or loop of Henle or whatever and another one. I mean, would they be different at all? Because it's essentially, I look at it as kind of a perfectly repeating structure. So I wonder whether
Actually there is a difference between the upper and lower glomeruli, but I don't know how much we know about the amount of variation, and there are even sex differences between those 2 regions, as I recall it. Maybe Mark knows more.
You're right. It's known there are differences between glomeruli towards the outer cortex and towards the inner cortex. And if you were to if you were to go to the Human Protein Atlas and look at the proximal convoluted tubule or distal convoluted tubule, you will see expression differences between adjacent cells that are both epithelial cells of the proximal convoluted tubule or distal. Is that a cell state? Is that a different cell type? I wrote, it's kind of philosophical how you define a cell, and some people like to split them into very particular groups. You can flow sort lymphocytes into 20 or 30 groups, or you can say they're all lymphocytes and call it a day. So back to that original point, it's hard to say where you stop, where something is a discrete cell type and where something is a state of a cell type. And that's just a great question for the Human Cell Atlas in general, and how we define that based on genetics or protein expression or histology or whatever that is, it probably all goes together, but that is, to me, one of the key points that this group will eventually solve for us.
Or location.
Or location.
Yeah.
But if it's the same, I mean, that's what I was just asking. Like, if the cell is really identical, it's absolutely identical in every sense: morphology, RNA, protein, you know, electron microscopy, structure, everything. But it's it's repeated in different locations. Then, you know, I would just call it the same. But I would definitely split B and T cells. I mean, that's whew. I think what we need is, so what I would love is to have as detailed information as possible, but organized, you know, to be accessible in a hierarchical way. So to satisfy both the lumpers and the splitters. I think that would be ideal.
So I pasted, and feel free to delete the image if it cannot be shared. So I took the liberty to paste the screenshot from EMBL into the chat log. So please do delete it if it shouldn't be there. But I think it's interesting to understand that that is actually very similar to what we are using inside of HubMap. So I pasted this right underneath. And then it's also, again, multi-level in the paper that Griffin discussed briefly. And then what I'm really looking for is these 3D objects, which are nested structures. So here you have this, just the object file loaded into the sandbox from Babylon.js, and then you can explore it, and you can click on certain subparts, and you will get to see the name for those. And you can, of course, turn them on and off. And I think, again, these kind of 3D models, it would be great to have more of those and freely available.
I mean, sharing the slide is fine with me. It's done by an illustrator who works in my group. Jana Elliott.
Would you like to put the name of the illustrator next to it?
Yeah, I'll add that.
And then we have, last but not least, a very, very important question, which is near and dear to my heart, which is taxonomy, nomenclatures, and standardization. So when Amy Bernard showed me what they have been doing for cell types of the brain, I was very intrigued, and I was very, very interested to then get similar clarity and standardization for the anatomical structure names and definitions, and the microanatomical structure names and definitions, and the cell types names and definitions that we are dealing with in the kidney. And then I was told that this doesn't exist. And so, yeah, so I'm not very happy about that part right now. But I would love to understand if there is more progress across other consortia. And if there is, I would love to understand how it was done, what kind of incentive structures, carrots and sticks you used to get that done. And ultimately can be shared and can we all benefit from it. And maybe it again takes that we do it for 5 to 10 organs and we agree on it and we agree on a process of how to get there. And then it becomes a de facto standard. And so if you know of other efforts to try to give us taxonomy, nomenclatures, and standardization, I would love to see more pointers in this green box in our meeting notes. And then
oh yeah, so there's one, I mean, you're probably aware of the Richard Shireman ontology paper.
Maybe not. If you could put that in, that would be wonderful, Sarah.
What do you mean, put into the Google Doc or put into the chat here?
Ideally, everything ends up in the Google Doc. So if any one of you has pasted something into the chat that they would like to see preserved, please do copy it over from the chat into the Google Doc, because my understanding is that the chat log is going to be deleted and is not going to be available at the end. So please make sure that everything that needs to be transferred is transferred back.
So while we have this moment, Jim, is there anything from what you were supposed to be leading that you wanted to bring up as well, since you had the group that sort of disappeared earlier?
So some of those, some of the themes and topics were I see documented in the Google Doc here. I don't know if it's immediately relevant to what Katy was talking about, although I guess in terms of what she's requesting, I'm just naively assuming that each of the communities are developing their own taxonomies and that are relevant to their organ system and even diseases of interest and so on and so forth. I know that those of us who are working on the brain, there's some standard ones that folk are just trying to make coherent and portable across the definitions. But in terms of part of how this is particularly relevant is that within, interestingly enough, within the brain cell census network, where we're just only focusing on the brain. And in this case, there was a network-wide decision to standardize around the Allen CCF. And you would think that problem solved because we just adopted a CCF as a standard for all consortia partners to use in describing their acquisitions, their output. It turns out that even within that definition, those of you familiar with Allen's work is that they've actually produced a series of CCFs. And so there are groups who are using the older version and others who are using the newer version. The consortia is supposed to standardize around the very latest version. And there's significant controversy in terms of the fact that even within the same, in this case, the same enterprise, the CCFs do not correspond. And this gets back to some of the definitions that certain structures that are defined the same name that are given across different CCFs were actually defined very differently in different versions of the different versions of CCF. So localizations done in one actually don't map into the into the same region in a newer version of CCF or a different version of CCF. Those are the kinds of things that we're trying to document and capture in some way, so that presumably we're far off from some universal CCF, and that we will be interoperating between different CCFs, and so any means of trying to standardize across CCFs would be extremely helpful toward that end, or at least minimum being able to understand what the differences are if you can't standardize, so that when you're talking about this structure, this region, and this CCF, you have to translate that into perhaps a different, a different part of the body in a different version. I don't know if other consortia are dealing with it. I mean, within our own brain initiative cell census network, we are trying to address this. And in fact, as part of the larger effort, one of the outcomes one of the outcomes would be yet another definition for many of the brain regions that are being studied, now using a consensus approach in terms of defining that particular region. Sorry, Katy, I had just taken over while you stepped out. Mark took the opportunity to to ask a question in terms of the subtopic that I was originally supposed to be leading. Mark, I don't know if I addressed what you were interested in hearing. I'll hand it back to you, Katy.
Yeah, and I love to see the meeting notes also. Thanks to the scribe, and also thanks.
Can I ask a question? I think that this is a major challenge, and I wanted to learn from the brain experience. What do you think leads to this sort of fragmentation? Obviously, it makes it so much harder to have a unified map if everybody else is referring to it in different ways or uses different reference systems. Is it everyone thinks that their system is better than the other ones, or they all started a while back and they don't want to change? I mean, anything that you can share.
In the case of the BICCN, it's actually very straightforward. I think it's just science marching along. And in this case, like I said, even though that we had standardized around the Allen series of CCFs, within the Allen organization, the series of CCFs they've defined have been based on different source materials. And with each new generation, they believe that they're coming up with quote unquote better definitions of motor cortex, because they're using more information to define the boundaries of that region.
Weigh in at all on this or no?
Yeah.
I think so. One point of clarification too is that James is talking about the mouse. So right now, the mouse reference atlas has undergone many different iterations, partly to get to a greater level of resolution that's down to the micron level. We pin things very, very, very specifically down, pretty much close to the cellular level. And that's been one of our goals for that mouse reference space, so the mouse CCF. The human is actually in a much more naive form. And I just posted both to the chat as well as to the Google Doc, the most recent release that we have for anatomical structures for not a human reference map we've annotated it at the Allen, but it's basically working with the MNI space that already exists. So that's working with a common reference space developed through Montreal Neuroscience Initiative that we've now layered on an annotation that's been done by one of our neuroanatomists, Songlin Ding. And that's basically what we're working with now for human. So human is actually at a very naive state comparatively. And I've been trying to work with Katy very, real time at the sort of leading edge of trying to get this information into a space so that it's just informing both the HCA as well as HubMap that we're working with this in the format that it's at now. And what we're doing at the Allen is making sure that we essentially pin and associate the annotated origin, structural and anatomical origin of where we have single cells to a particular neuroanatomical framework based on MRI data from the source in which it was excised. So this is very specific to sort of our own workflow that's very much emerging in real time. But we're working collaboratively with whoever we can, most predominantly, as James said, with through the BCDC network, through BICCN as part of the BRAIN Initiative, but trying to, through these kinds of communication interactions, make sure that it's at least synced up awareness-wise with any other reference space that needs to be tied to it. So Ed Lean is one of the key people who I know has been very involved even in the arranging of this particular meeting, working through HCA, through everybody else. So it's, I would say for mouse, the iteration component is an issue, practically speaking. For human, it's different. It's we're really at the bleeding edge, and we're if anything behind the curve in terms of the fact that the human brain is so complex that our ability to annotate at the structure that we want to annotate for single cell resolution mapping is not there. So we've actually done it for part of the cortex, and I've provided Katy with that grid, that sort of 10 by, it's not the 10 by 10, it's the other grid, the 6-point grid. I forget which grid it is, but it's a grid that starts mapping sort of fine to coarse anatomical referencing of single cell origins to their parent structure. That's a taxonomy based mapping. And then the spatial mapping is something that's much harder to do, and we haven't tackled that at all. But that's sort of in a quick summary of what the Allen is doing now.
Thanks for the clarification, Amy. I completely, and yes, I completely neglected to mention that we were focusing largely on the mouse. And so there, and this touches on, I think, an earlier comment that was made and that Amy has highlighted in the human brain. There's not only addition of complexity, but there's also this variable that was mentioned earlier in terms of variation. So in the mouse animals, these are specific strains. Obviously, we're not doing that for the human brain data. We're not breeding humans that we're subsequently characterizing. So there's that additional dimension that's really extremely challenging. That was pointed out earlier.
And I can tell you that I was actually envious because the brain has been known and mapped, of course, not a single cell level, but for 100 years. I mean, people seem to agree on a lot of the spatial information regarding the brain. Some other
Now, I have
tissues
to
are
just
not.
butt in and say that that is not quite
Not
accurate.
true? They don't agree, you mean?
No, absolutely not. Absolutely not.
Why am I not surprised?
At the level, there's a cerebellum, there's a hippocampus, but as soon as you get down into any really detailed spatial mapping, that's exactly where there's a lot of iteration. And certainly if you're looking across human and wanting to do anything in human where you're now mapping actual data, there's huge variation. So just the structural anatomical variation, it's massive. And if you're looking at any kind of developmental axis or any kind of clinical involvement, there's just, I mean, like, it's so widely different. It's like everyone is its own unique thing. So at a very high level, I made this mistake actually with an immunology comparison too. I'm like, oh, everybody knows all the blood cells. Like, no, it's evolving all the time. And I think that very much our understanding of the anatomy is just reiteratively shaped by the data and the data availability. So these things are so iterative, even for something for which people think there's some resolution.
So,
Sorry
but that's
to
good to
interrupt.
know. It means that's a evolutionary pain that we have to go through as we get more and more precise. We get less and less unite in what we think we are looking at or positioning, right?
But I think the beauty is that we are now having potential solutions that could map something that looks like a textbook, these kind of 3D models that probably many of you have learned in your biology classes. And then you look at something that Kristen gave us from the visible human, and this is from one person, and it's very different, left and right, totally different. You can look at it from the other side, and it's not like that textbook, right? So on our end, we have been debating which one to show in the registration user interface. Obviously, everybody wants to see this. This is easy, nicely organized. You can just put it here if you cut it out of the renal pyramid. If you have something like this, it might not even show exactly where those overlapping renal pyramids are. So it's for us a really interesting discussion how to deal with that human diversity. And I think few of us have a good understanding of how diverse we all are. Maybe it's only the pathologists and anatomists among us which really understands that truly. Still, I still remember that textbook version. And the beauty is, however, we can use that textbook version. We can give it to people as a registration user interface. And then if the vasculature approach works, or some other approaches that others are working on, I know Rahul has his own ways to map one lung into another lung. For those purposes, then they can then take that textbook and map it over to the real thing and the other way back. So I think as soon as we have a mapping that works across individuals, then of course that mapping would also work to move it over to a textbook version or to move it over to a very conceptual understanding of how the human vasculature, for instance, works. So we have these multiple projections across individuals, across textbook work, and across very abstract, but potentially even more valuable conceptual maps, which we could use as base maps. And as some of you know, much of our work here at CNS, the center I direct, is on large-scale maps of science. And those also don't exist. They are artifacts, they are semantic spaces that we use to understand where the landscape of biology is, and engineering and mathematics, and how they all interlink and feed each other and help each other. And similarly here, we could see the human body as a system that needs to be given a shape, even though it already has the anatomical shape, but potentially a different shape, a different reference system is actually much more beneficial for certain types of questions. And then we can use that reference system to overlay disease, to overlay evolution, to overlay what happens as we age. I think many of us are interested in that part, and HubMap is dealing with different ages. We are not dealing with disease, and we are not dealing with anything before 18. So I think it will be interesting to see how we can agree on certain reference systems for certain organs. And again, that table, just having the names of those major areas that people care about, and volumes here in this case that people care about, and how to refer to them is incredibly valuable. And then again, it's a nested structure. It's celled inside of functional tissue units, inside of microanatomical structures, inside of larger and larger, and ultimately the human body. So I think if we could agree on these initial tables, I would find that very valuable. I wanted to make sure that everybody got a chance to put their name at the end of the document. So I tried to fill out some more of those sections, and you're all welcome to also look at all these sections one more time. I think we have 7 more minutes before we get to hear our bravo. You're done. I think
Katy, I want to make a quick note that I am probably one of those lumpers, maybe because I started by being a physicist. I believe that there are a few principles that are very important, and the rest are small variations around the principles. And when you are completely off the principle, obviously you have disease. So I think the variations and heterogeneity, some of it is normal. So between, say, the organs of a female and a male, there is a difference, right? But it's a normal difference. And then when you go at the meso scale, you would have probably differences in the vasculature, maybe some people, or the lung of the trachea or branches. Some people have 3 and some people have 4. But if they don't have functional consequences and they all behave normally, when you go at the smaller level where everything has to work perfectly, everybody's functional units contain the same type of cells and most likely about the same number of cells. And if they don't, that means they're not normal. So I think there are certain patterns and certain principles and rules that can help us build something that looks like the normal human, although we realize there will be variation up and down, but when the variation becomes abnormal is probably when you find disease. Sorry, I philosophically speaking, I guess.
I think I captured this in our meeting notes here.
Katy, with the last few minutes left, one of the things I think the organizers asked is if we wanted any kind of follow-up activity to act on the outputs of this breakout session?
So I think as organizers, we had discussed that we have a kind of debrief after all of this is said and done. And I think there are a number of really interesting sessions tomorrow also, which I would love to know more about before I have the debrief. I would love to see if people want to come back to a 1-hour debrief. And I tentatively put in here that we might all be available, fingers crossed, probably not, but fingers crossed on April 9, and we can find other times also. But I think there will be a list of names and email addresses at the very, very end of the document. So again, please make sure that your name and your email address is there, because we will have somebody who knows how to schedule very busy people try to find a time that most of us can do it, maybe not all of us. And it would be nice to really see what we can truly do together. And I think then there are a number of one-on-ones I would love to have. And so I will reach out personally to just schedule those, and feel free to do the same. I think it's not always the most productive to all meet all 20 of us. Please do reach out to the people you want to connect with. And then let's try to schedule one Zoom debrief for all of us around April 9.
Katy, do you want to reach out to me?
Yes.
We can schedule one, because I'm like over the Easter period, I'm working the whole time actually. So.
Okay. Yeah, no, I would love that. And I have your email address here.
Fantastic. Yeah.